Have you ever looked at someone and wondered whether their family history tells the whole story? For generations, the Métis people have carried an identity unlike any other in North America. They are often described as a bridge between two worlds, yet modern genetics reveals a story far more complex than anyone imagined. Hidden within their DNA are centuries of survival, migration, love, conflict, adaptation, and resilience. Every chromosome carries fragments of a journey that reshaped an entire continent. Today, scientists can read those fragments with incredible precision. They can compare genetic markers, identify ancestral origins, and trace family connections that were invisible just a few decades ago. But the results do not simply confirm old stories. In many cases, they challenge them. What exactly makes Métis genetics unique? Is it possible for DNA to distinguish culture from ancestry? And why have researchers become increasingly interested in studying one of North America's most misunderstood populations? The answers begin more than 4 centuries ago. During the early 1600s, European explorers arrived across vast regions of what is now Canada. French traders, Scottish merchants, and later British settlers entered territories already occupied by hundreds of indigenous communities. The newcomers depended heavily upon indigenous knowledge for survival. They needed guides, they needed interpreters, they needed people who understood rivers, forests, weather, wildlife, and trade routes stretching thousands of kilometers. Relationships slowly formed. Some were based on mutual respect. Others developed under difficult historical circumstances. Families emerged. Children were born into two different worlds. Unlike many colonial societies, where mixed ancestry children were forced to abandon one side of their heritage, many communities across the Canadian interior created something entirely new. Those children did not simply become European. They did not simply remain members of one indigenous nation. Instead, over generations, a distinct people gradually formed. That people became known as the Métis. Today, genetics confirms that this history left measurable biological patterns. Yet DNA tells only part of the story. Scientists repeatedly emphasize that being Métis is not determined solely through genetic ancestry. Culture, community, language, history, family traditions, these remain essential. DNA can reveal ancestors. It cannot define identity. That distinction is one of the most important lessons emerging from modern genetic research. For decades, researchers believed populations with mixed ancestry would simply blend genetically until their original signatures disappeared. The Métis challenged that assumption. Instead of becoming genetically invisible, many Métis communities developed recognizable ancestry patterns. Through generations of families marrying within neighboring settlements, population geneticists call this founder effect. When relatively small communities expand over time, specific genetic variants become more common than expected. This phenomenon has been observed throughout human history. It appears among Icelanders, among Ashkenazi Jewish communities, among the Amish, among Finnish populations, and researchers discovered similar patterns in several Métis settlements. That discovery surprised many scientists. It suggested that history leaves fingerprints inside our DNA. To understand why this happened, researchers first examined family trees. Historical records from the Red River region revealed something remarkable. Certain founding families appeared repeatedly across multiple generations. As descendants continued building communities together, pieces of shared DNA became increasingly common. Modern genomic analysis confirmed those observations. Although individual ancestry remained highly diverse, communities developed distinctive genetic characteristics reflecting their unique history. This does not mean every Métis person shares identical DNA. Far from it, Métis populations remain incredibly diverse. Some families have predominantly French ancestry. Others trace paternal lines to Scottish fur traders. Many inherit maternal ancestry from Cree, Ojibwe, Saulteaux, Dean, or other indigenous nations. Each family carries its own remarkable story. Genetics simply provides another way to observe that complexity. Scientists frequently analyze three major categories of ancestry. Autosomal DNA represents genetic material inherited from both parents. Y chromosome DNA traces direct paternal ancestry. Mitochondrial DNA follows direct maternal ancestry. When researchers compared these systems across historical Métis families, fascinating patterns emerged. Many paternal lineages reflected European origins. French surnames appeared repeatedly. Scottish family lines also became prominent. Meanwhile, maternal mitochondrial DNA often showed indigenous ancestry. This pattern perfectly matched historical records, describing relationships between European traders and indigenous women during the fur trade era. Independent lines of evidence suddenly reinforced one another. History matched genetics. Genetics matched history. Few populations illustrate this relationship as clearly as the Métis. However, scientists quickly encountered another challenge. People often assumed DNA could identify someone as Métis with a laboratory test. That assumption proved incorrect. There is no single Métis gene. There is no exclusive Métis chromosome. There is no biological marker capable of defining cultural identity. Instead, researchers study probabilities. They compare genetic variation across thousands of locations within the genome. Patterns emerge across populations. Individuals remain unique. This distinction matters enormously. Modern genetics helps reconstruct history. It should never replace community recognition or personal identity. One of the biggest breakthroughs occurred when researchers began sequencing entire genomes rather than examining only small portions of DNA. Earlier studies analyzed relatively few markers. New technology examines millions simultaneously. The results became dramatically more detailed. Scientists could estimate ancestry proportions with increasing accuracy. They could identify historical population movements. They could reconstruct migration pathways. They could even estimate when different ancestral groups mixed. For many Métis families, those estimates aligned remarkably well with written historical records from the 18th and 19th centuries. Another surprising discovery involved genetic diversity. Many people assume mixed ancestry automatically produces enormous diversity. Initially, that is often true. However, after multiple generations within relatively connected communities, unique genetic signatures begin emerging again. This creates fascinating biological patterns. Researchers studying Métis populations observed combinations of Indigenous and European ancestry, unlike those seen elsewhere. Not because the DNA itself became entirely new, but because centuries of shared history produced distinctive population structures. Every generation added another chapter. Every family contributed another branch. Every birth expanded a remarkable genetic mosaic. Yet perhaps the most important discovery had nothing to do with ancestry percentages. Instead, it concerned health. Modern medical genetics increasingly recognizes that population history influences disease risk. Certain inherited conditions appear more frequently within some populations than others. Understanding those patterns helps physicians improve diagnosis, screening, and treatment. For decades, indigenous communities remained significantly underrepresented within genomic research. The same was true for Métis populations. Without sufficient genetic data, medical researchers struggled to understand inherited conditions affecting these communities. That gap created real healthcare challenges. Researchers gradually partnered with Métis organizations to address this imbalance. Community consultation became essential. Respect became central. Consent became non-negotiable. Unlike earlier eras when biological samples were sometimes collected without meaningful collaboration, modern studies increasingly emphasized shared decision-making. This shift transformed genomic research. Instead of studying communities, researchers began working alongside them. Trust slowly developed. As participation increased, scientists identified important health-related genetic patterns. Some involved metabolism, others involved cardiovascular health, still others related to rare inherited disorders. Each discovery carried potential benefits: earlier diagnosis, better prevention, more personalized healthcare. But every scientific advance also raised ethical questions. Who owns genetic information? Who decides how DNA samples are used? Can ancestry testing companies accurately represent Métis identity? Should governments rely upon genetics when determining indigenous status? Most researchers answer that final question with a clear no. Identity cannot be reduced to DNA. Community matters, history matters, culture matters, family matters. Genetics contributes valuable evidence. It does not replace lived experience. This lesson became especially important as commercial ancestry testing exploded worldwide. Millions of people mailed saliva samples hoping to discover hidden family secrets. Many expected simple answers. Instead, they received probability estimates. Desorientages changed whenever databases expanded. New reference populations altered previous results. Algorithms improved continuously. DNA testing became more accurate. Yet it also became more complicated. Someone might discover indigenous ancestry several generations back. That finding alone does not automatically establish Métis identity. Likewise, someone raised within a recognized Métis community might inherit fewer detectable indigenous markers purely through random genetic inheritance. DNA follows biology. Identity follows history, family, and community. Confusing those concepts creates misunderstanding. Scientists work carefully to explain this distinction. Meanwhile, historians continue uncovering remarkable stories preserved within archival records. One famous example comes from the Red River settlement. Throughout the 19th century, families recorded marriages, baptisms, land ownership, and community events. These documents now combine with modern DNA analysis to reconstruct family histories in astonishing detail. Researchers sometimes identify living relatives separated for generations. Entire family branches reconnect, historical mysteries disappear. Forgotten ancestors return to memory. Technology becomes a bridge connecting present and past, yet not every story ends neatly. Some records vanished, others were destroyed. Names changed, languages shifted, political boundaries moved repeatedly. DNA sometimes fills those historical gaps. Sometimes it raises even more questions. One family may discover unexpected Scottish ancestry. Another uncovers French origins previously unknown. Another identifies connections to multiple indigenous nations across different generations. Every genome contains surprises. No two stories are identical. And perhaps that diversity explains why the Métis remains such an extraordinary focus for modern population genetics. Their history refuses simple definitions. Their ancestry refuses easy categories. Their DNA reflects centuries of human relationships far richer than any single label can describe. Scientists continue sequencing genomes. Historians continue opening forgotten archives. Communities continue preserving oral traditions. Together, these three sources create an increasingly complete picture. A picture not defined by percentages, but by people. A story not written solely inside DNA, but carried through families determined to remember where they came from. As DNA sequencing became more advanced, researchers started asking a new question. Instead of simply asking, where did these ancestors come from? They began asking, how did generations of shared history shape the genomes of modern Métis families? The answers were extraordinary. Every child inherits half of their DNA from each parent. With every new generation, chromosomes are shuffled through a process called recombination. Pieces from grandparents, great-grandparents, and countless earlier ancestors are mixed together in unique combinations. No two people inherit exactly the same genetic mosaic, not even siblings for Métis families. This process created something remarkable. Over centuries, Indigenous and European ancestry became woven together in patterns that reflected real communities rather than random mixtures. Those patterns tell a story of families who lived, worked, married and raised children together generation after generation, the fur trade was more than an economic network. It became the foundation of entire communities. Along major rivers and trading posts, families established homes that eventually grew into thriving settlements. Children learned multiple languages. Many spoke French, many spoke Cree. Others learned English or Ojibwe. Some even spoke Michif, the unique language that blended elements of French and indigenous languages into something entirely new. Language, like genetics, became evidence of shared history. One cannot exist without people passing knowledge from one generation to the next. That same principle applies to DNA. Every generation leaves traces inside the next. Researchers examining historical migration discovered that many Métis families traveled enormous distances across Western Canada. They followed rivers. They established new settlements. They adapted to changing political boundaries. Despite these movements, extended families often remained connected through marriage and community traditions. This explains why scientists sometimes detect shared genetic segments among families living hundreds of kilometers apart. Those shared segments act like biological echoes from common ancestors. The larger the shared DNA segment, the more recent the shared ancestor is likely to be. Smaller segments usually point to relationships much further back in time. Modern software can estimate these relationships with surprising accuracy. Combined with historical records, researchers can reconstruct family histories spanning centuries. But the story is not only about ancestry. It is also about survival. History brought epidemics, displacement, conflict, and changing governments. Many Indigenous and Métis communities experienced tremendous hardship. Every generation that survived passed both culture and DNA to the next. That resilience is visible in family histories across Canada today. Scientists are careful not to describe resilience as something encoded in DNA. Resilience come people, communities, traditions, and choices. DNA records ancestry. People create history. Another fascinating area of research involves inherited health conditions. Population genetics helps identify rare genetic variants that may occur more often in certain communities because of historical founder effects. This knowledge allows doctors to improve screening programs and offer earlier diagnosis for families who may benefit. Importantly, these discoveries are made through partnerships with Métis organizations and community leaders. Modern ethical research depends on informed consent, transparency, and shared decision-making. The days when scientists simply collected samples without consultation are increasingly viewed as unacceptable. Communities deserve control over how their genetic information is used. This principle has become one of the foundations of Indigenous genomic research. DNA is deeply personal. It connects individuals to parents, grandparents, and ancestors. It should never be treated as merely another scientific data set. As consumer DNA testing became more popular, many people hoped a simple saliva test would answer lifelong questions about identity. Sometimes the results revealed surprising family connections. Other times they raised unexpected questions. Someone might discover French ancestry. Another might find Scottish ancestry. Others might identify Indigenous ancestors several generations back. These discoveries can be meaningful. But researchers continue emphasizing one essential truth. Genetic ancestry is not the same as cultural belonging. Being part of a Métis community involves history, family relationships, shared traditions, and recognition within that community. A laboratory cannot measure those things. DNA can identify biological relationships. It cannot define who someone is. This distinction protects both science and culture from being misunderstood. Meanwhile, advances in genome sequencing continue at an astonishing pace. Scientists can now analyze millions of genetic markers across an entire genome in a matter of days. Artificial intelligence helps compare enormous databases while protecting personal privacy. These technologies allow researchers to identify migration patterns that would have been impossible to detect only 20 years ago. Every improvement adds another piece to the historical puzzle. Yet many questions remain unanswered. Researchers continue studying how different Métis communities developed unique genetic profiles. They are learning how geography influenced marriage patterns. They are examining how population size affected genetic diversity. They are investigating how inherited traits changed over generations. Each study contributes another chapter to a much larger story. One lesson appears again and again: human history is far more connected than people once believed. Families crossed rivers, mountains, and political borders. Communities exchanged knowledge. Cultures influenced one another. Genes moved alongside people. The Métis story illustrates this beautifully. It reminds us that history is rarely simple. It is made of countless individual lives. Each family carried hopes, fears, traditions, and dreams. Each child inherited more than DNA, they inherited stories. Today, many young Métis people are reconnecting with those stories. Some are learning traditional languages. Others are exploring family genealogy. Many are working with elders to preserve oral history before it disappears. At the same time, researchers continue refining genetic tools that help families understand biological ancestry with greater accuracy. These two paths, science and tradition, are not opponents. When approached respectfully, they complement one another. Science explains inheritance. Communities preserve identity. History connects both. Perhaps that is the greatest lesson modern genetics has revealed. DNA is powerful. It can uncover forgotten ancestors. It can identify distant relatives. It can help doctors improve healthcare. It can illuminate migration routes that remained hidden for centuries. But DNA cannot replace memory. It cannot replace language. It cannot replace belonging. Those things live within families and communities. The Métis remind us that identity is built from far more than chromosomes. It is built from relationships, from shared experiences, from resilience passed through generations. Every genome tells part of the story. Every family tells another part. Only together do they reveal the full picture. As genome research continues into the future, scientists hope to answer even more questions about ancestry, health, and human migration. But perhaps the most important discovery has already been made. Behind every sequence of DNA is a human life. Behind every chromosome is a family. Behind every family is a history worth remembering. And for the Métis people, that history continues to inspire researchers, historians, and future generations alike, because the greatest discovery was never hidden inside a laboratory. It has always existed within the people themselves, their culture, their families, their resilience, their identity, and their extraordinary journey across history.